Complex permanent tissues. Complex tissue is defined as a group of dissimilar cells that performs a specific function collectively. Complex permanent tissues are of two types: xylem and phloem. These tissues are helpful in conducting water, minerals, and inorganic substances in the plants. Hence, they are also known as vascular tissues. Xylem. This complex tissue is used to conduct water and minerals to the aerial parts of the plant from the roots. It is also called hydrome. Xylems is made up of trachyary elements, xylem fibers, and xylem parenchyma. Trachyary elements. They conduct water and minerals in the upward direction. These cells have lignified secondary walls and contain lumen. Trachyary elements are further divided into two types: tracheids and vessel members. Tracheids. Tracheids are elongated cells that have broad middle region and sharp tapering ends. The cell wall is thick and lignified with bordered pits. These are dead cells since they have no protoplasm. Tracheids help in conduction and provide mechanical strength. They have arranged in parallel rows and have vascular bundles. Vessel members. These cells are elongated and wide. Their cell wall is thick and lignified. These are dead cells. Vessel members are also called perforation plates since they have oblate and perforated end walls. The area where the vessel members join is known as vessels or tracheae. The vessel membrane with a single perforation in the cell wall is called a simple perforated plate, whereas the one with many perforations is called multiple perforation plate. The secondary thickening of the cell wall formed due to the deposition of lignins is of different types. Angular thickenings, that is a ring-like thickening. Spiral thickenings that have continuous helices. Scalariform thickenings that has ladder-like thickenings. Reticulate thickenings that are like a network. Pitted thickenings that comprises of pits. Xylem fibers. Xylem fibers are elongated and spindle-shaped. These cells have sharp ends and consists of a thick and lignified cell wall and lumen. They provide mechanical strength to the vascular bundles. Xylem fibers are of two types: libriform fibers and fiber tracheids. Xylem parenchyma. These are parenchymatic living cells with thin walls and has vacuoles present in the cytoplasm. The cell wall is non-lignified and flexible. Primary xylem contain axial parenchyma that is arranged parallel to the axis. Secondary xylem contains both axial parenchyma and ray parenchyma in vertical and horizontal rows respectively. These cells are useful in the storage of starch or fat. Phloem. Phloem is the vascular tissue that transports sugars from source tissues, that is, leaves, to the other parts of the plant. Phloem is also called leptome. Phloem consists of sieve elements, companion cells, phloem fibers, and phloem parenchyma. Sieve elements. Sieve elements are the main conductive cells of the phloem. The nucleus here disintegrates on maturity, and hence we say that sieve elements are anucleate. It consists of a vacuolated protoplast. Sieve elements may be divided into two types, based on the sieve areas in their cell walls, that is, sieve cells and sieve tube members. Elements with unspecialized sieve areas are the sieve cells, and each sieve area consists of minute pores called sieve pores. Highly specialized sieve areas that are organized into sieve plates are known as sieve tube members. These are arranged vertically one above the other through cross walls that are perforated to form sieve plates. Companion cells. These are closely related to sieve tube members. They consist of a large nucleus in the dense cytoplasm. Phloem fibers. Phloem fibers are the dead cells of phloem. The cell walls are thick and lignified. They may consist of a simple cell wall or have bordered pits along with lumen. They give mechanical strength to the vascular bundle, phloem parenchyma. These cells are thin-walled and consist of vacuolated protoplasts and a large nucleus. Its cytoplasm is rich in starch, fats, tannins, and resins. The primary phloem consists of only axial parenchyma, while the secondary phloem consists of both axial and ray parenchyma.